convolutional neural networks. So for the first part of this talk, lecture, I'm actually going to be going over a bit of the history, where exactly these models came from. How did we arrive at the models? The models were not originally arrived at by assuming that you were just going to be scanning. Is this too loud? Better now? So they were not originally arrived at by assuming you're going to be scanning the input. The reasoning was more biological, and we'll go over that, and then we'll go over some of the, we'll sort of revisit ConvNets in a little more detail. So the story we've seen so far is that pattern classification tasks like, does this picture contain a cat, or does this recording include the word hello, are best performed by scanning for the target pattern. And scanning an input with a network and combining the outcomes is equivalent to scanning with the neurons individually and then working on the scanned outputs of the neurons. We saw that deformations in the input can be handled by max pooling, which, as we'll see today, there are other ways of doing it. And we finally saw that when you're scanning two-dimensional images or higher dimensional images, we often call it a convolutional network, ConvNet for short. And if it's just a scan along time, if it's one-dimensional, it's called a time delay neural network. So a history. It all began with people trying to understand vision. What do animals see? What's the neural process from the eye to recognition of an object? So in these figures over here, does anybody see a cube in the top left corner? Everybody sees a cube. Is there a cube? No, right? All you've drawn is a bunch of those hokey hippie circles, and it actually looks like a cube. Same thing over here. What is that? Is there a dog? Is it really a dog? Not really, right? So there's this business of Gestalt. The brain has an innate tendency to organize disconnected bits into whole objects. And a lot of the early research on human vision or animal vision was based on such behavioral studies. What are the kinds of illusions that we can cause? And how do animals and people interpret visual scenes? But there was no real understanding of how the brain processed the actual images that came in. So the first real work on this was reported by Hubel and Wiesel in 1959 on this very nice paper called Receptive Fields in the Cat Striate Cortex. So the way the human brain is designed, we have the eye, and that's my eye. And right at the back, this is you would have the retina. So the retina is a large, somewhat circular surface that the images fall in, and it's sort of dotted with, it's coated with neurons, and these neurons all connect into the brain. That's my figure for the brain. So actually it turns out that the optical neurons that go into the brain are just an extension of the brain. In fact, your eye is a part of your brain, amazing as it is. So when light falls on the eye, these images are first conveyed directly to somewhere in the back of the head, the region called the V1, the V1 region of the visual cortex, the portion of the brain that actually processes vision. And this portion of the brain has a striated structure, and in the case of cats. And so they spoke of receptive fields in the cat striate cortex. This is what they were trying to analyze. They worked on 24 poor cats. They anesthetized them, they immobilized them, they anesthetized them with truth serum, and then they held their irises open. So the poor things, now of course they're immobilized, they're anesthetized, and they can't do anything about it. And then they beamed little portions of light into the little beams of light into the cat's eye, and studied the response. So the way they did it was they would stick electrodes into that looked at specific neurons in the cortex. And that specific neuron as it is, it's not just connected to one cell over here. This neuron is actually connected to a region of your retina. Now, if the light falls on all of these portions of the retina that it's not connected to, the neuron is obviously not going to fire. But even within this region that it's actually connected to, it turns out that there are two kinds of responses that you get. You have regions where if light falls on these regions, this actually kills the response of that particular cell. So if there's any light on these inhibitory regions of the retina, then the neuron will definitely not fire. And on the other hand, you have these other regions where if light falls, the retina, that cell is going to fire with high energy. So the overall, now what they were really trying to study was what was the immediate response of these retinal cells. As soon as the light falls on the retina, how does these, what is the response within 20 milliseconds out here? So what they found was that first, these guys don't respond to everything. There are specified regions on the eye that each cell responds to. This was what they call the receptive field of the cell. So this is where the terminology comes from. This is the receptive field of the specific cell in the brain that is one of the cells that's trying to interpret images. Secondly, you had these inhibitory regions where if light fell, the neuron definitely wouldn't fire. And then you had the excitatory regions which cause the neuron to fire. Now, if you just beamed a diffuse light on this entire region, what would happen is these inhibitions would cancel out these excitations, nothing happened. So what it turns out is that the light must fall. The patterns of light that the cell responds to are patterns where there is no light over here and there is light over here. So what does that pattern look like? A very distinctive line. So here are the kinds of responses that they actually found. This was mice, monkeys, these are all later research. But basically, if you sort of took a spot of light and bounced it around on the retina, then what would happen is that if the light, where are my TAs and can someone monitor Piazza please for me? If Roshan? Thank you. Anyway, so if you sort of moved the light along the retina, then you would get a response. And what specifically they found was that these receptive fields had nice oriented vertical or oblique patterns. So here was some of the results. This was one of the neurons. If they took a light and moved it sideways, the neuron didn't really fire. If they moved it along this angle, there was some firing. As they increased the angle at which they were moving the light, they found in this particular neuron that when the light is being moved up and down in a vertical direction, the neuron fires most. In other directions, the firing goes down. When it's horizontal, the neuron doesn't fire at all. So there's a very nice oriented arrangement to each neuron seems to be responding to very specific directions of excitation. So oriented slits of light were the most effective stimuli for activating these cortex neurons. Now, this is not the interesting portion for us. This is just an aside. What they found later was that the cortex consists of two levels of processing. Why exactly does this cortex respond to a oriented pattern of light? So the way it did it was this, that if you looked in this region of the retina, all of these guys, you'd have many cells over here, and all of these would connect to the particular cell in the striate cortex. And so the cell would respond if all of these guys fired. In other words, it was responding to oriented directions of light. But then something happens in the first layer. This kind of sensitivity, you're just looking for light that follows a specific pattern, and this kind of response can be noisy. So what they found was that there were actually two levels of cells in the brain. So you had some cells out here which would respond to this orientation of light. But then these guys were kind of sensitive to noise, so they could be fooled. But then the brain does a cleanup. So behind this, these they call the simple cells S. These S cells, the information from these S cells get fed backwards to another collection of cells called C cells or complex cells. And what the complex cell does is to look at a whole bunch of simple cells that is responding to a specific orientation, and it effectively votes over them. So what happens is that the complex cell does not sort of cancels out the response to noise that the simple cell has, and the complex cell is what gives you the final nice directed response to the input image. So they have this very nice structure. Complex cells build up their responses from similarly oriented simple cells, and also they found that there's a whole layer of in here you had the whole layer of layers of response. So you had simple cells which were cleaned up by complex cells, then you had more simple cells which were looking at the response of the previous complex cells, and those were cleaned up by simple cells, and so on. And so as you went on, you actually found that they found that more complex patterns were composed by composing early neural responses. So you could actually build up a cat, the image of a cat, through these little linearly oriented responses of the earliest layer of cells. They did a whole bunch of experiments, and each of them slightly more cruel than the other. So towards the end, they were working on live monkeys, which is not a nice thing to be doing. Anyway, but getting back to our story. And it turns out that some of the results were not even complete or maybe wrong, which is kind of sad. But coming back to our story. So here's a nice little model for how the brain kind of processes images. This was discovered in 1959, but then to come up with a computational model took a couple of decades. And that was by this gentleman here, Kunihiko Fukushima, who came up with the model of that we are familiar with today. Now, one of the things he found is he realized was something like this is not position invariant. So if you have a cell on the right side of my eye, it's responding to images on the left. But then, if this one learns to respond to my grandmother's image to the left, it's not going to respond to my grandmother to the right. On the other hand, it doesn't matter where my grandmother is, I'm going to recognize my grandmother. So this model doesn't explain the notion of grandmother cells. How many of you have heard of the Jennifer Aniston neuron? Really? So it turns out the neuroscientists have a great deal of fun out of epileptic patients. They open their skulls up because they have lesions in the brain, and they perform surgeries, but there's this brief period of time where these people are lying in this hospital where their skulls opened up and their brains exposed. So now they can begin running experiments. They put these little sheets of sensors on the brain, and they can figure out what the brain actually responds to. And they discovered that in one particular gentleman, there was a specific cell that only responded when he was shown pictures of Jennifer Aniston. So this is the Jennifer Aniston neuron. Anyway, each of us has a collection of these cells. You'll have a Jennifer Aniston neuron. You'll have whatever, any famous person, your wife, your mother, but there are specific cells that respond to these things. But here's the important thing. This cell responds, or your grandmother, regardless of whether Jennifer Aniston's up here or to the side. So how do we get this position invariance? And this is how Fukushima actually explained the whole thing. First, he sort of computationalized this little structure over here. he hypothesized that the visual cell system consists of a hierarchy of modules. Within each module, you have a collection of S cells, simple cells which respond to patterns, followed by C cells, the complex cells which clean up the response of the simple cells. This is again just building upon Hubel and Wiesel's discoveries. Then subsequently, the output of these complex cells are worked on by the downstream simple cells. So here's how he actually modeled the whole thing in terms of position invariance. So let's think of this as my neuron, as my retina. Then I might have a simple cell that is looking at this location. It's not actually looking at just one location. As we saw, a simple cell actually has a receptive field. It's looking at a small region in my retina. Then there is another simple cell which has exactly the same response as this guy, but it's looking at a slightly shifted version of shifted location in my retina. And then there's a third simple cell which has exactly the same response as this guy, which is looking at a somewhat more shifted version of shifted position. So there's an entire collection of these simple cells, all of which are identical, which together look at the entire scene that I'm viewing. Now, it's not enough to have one simple cell. We have many such simple cells. Each of these is looking for a different pattern. So, for example, I might have one which is looking for eyebrow-like patterns. I might have one that is looking for eye-like patterns. And each of these is going to be an entire collection of these cells which together look at the entire retina. Right? Yes? This was the hypothesis that Hubel and Wiesel came up with. This is Fukushima. There's some level of, it's the actual human visual system is way more complex than anything we're actually than our computational models. But as it turns out, if Pulkit can be here next week, I'll have him talk about it. Our computational models actually do a pretty good job of simulating what happens in the brain, which is pretty amazing. So either next week or towards the end of the semester when we have Mike Tarr, we'll actually look at this. It's pretty impressive. So anyway, but then as a simplified model, this kind of makes sense, right? But then it's not enough to have these simple cells. Following these simple cells is a collection of planes of complex cells. And what is the complex cell really doing? The complex cell, I have a complex cell which looks at a collection of simple cells, which is looking in a small region out here and taking a vote across them and giving you one response. So the output of the complex cells for each plane is actually a much smaller plane than the output of the simple cells itself. And so you get the idea, right? You're going to get one complex cell plane per simple cell plane, and you have as many simple cell planes as you have detectors. Now, and then you have an entire cascade of these things. So the output of these complex cells is now goes into the next simple cell. But here is the interesting thing. From each of these simple cells, I get one complex pattern, but when I get to the next layer of processing, this simple cell is not looking just at this one, but is looking simultaneously at all of these fellows. What is that? Why would you want to do that? So look at this figure here. Here, if this is your retina, then you have one plane of C's which is looking all over these guys to look for noses. Then I would have another one which is scanning all of these guys to maybe look for eyes. And then I have another one which is scanning all of these guys to maybe look for mouths. So if I want to find my grandmother, I must find a nose, a mouth, and an eye in the same place, right? So this guy, my grandmother detector, is going to be looking simultaneously at the same location in each of these three maps. Why simultaneously? Because this guy is looking at this region of my eye. This guy too is looking at the same region of my eye. And this guy too is looking at the same region of my eye, right? So by simultaneously looking at all of the maps, I'm sort of localizing a more complex pattern that I can compose from the simpler patterns that I got one layer down. And so you have an entire collection of these of these detectors, next level detectors, and they in turn have their own simplifiers, the cleaners, the C cells, and then you have the next level of S cells and so on. So you see how the whole model works, right? And the basic idea here, furthermore, he said, was that these simple cells, they do the job of detecting patterns. They can be learned. The complex cells, they have only one simple job. They're cleaning up the predictions of the simple cells. There's nothing to learn. It's like a max operation, right? So these S cells are plastic. They can be learned. And the C cells, their responses are fixed. And finally, in Fukushima's model, each S cell was looking at an elliptical region in the output of the previous layer. So this guy, for instance, the one who's looking for my grandmother, is simultaneously going to be looking at an elliptical region here, here, and here. And then finally, Fukushima came up with the final bit. He actually put the math to it. So what was the S cell's response? The S cell would respond to a collection of neurons over here, and the actual response, he gave this hideous-looking function. I'm not really sure how he arrived at the function, but that's besides the point. And then the C cells, how does it actually combine the evidence from a collection of S cells? For that too, he had a function. And fortunately for us, it turns out that this looks like a ReLU and this looks like a max. So problem solved, right? We can just use, so this also sort of explains some of the choices we will make when we actually implement these models, right? Now, how do you actually learn this entire structure? First, you observe that as you go through the network, the receptive field of each neuron gets larger and larger and larger. What do I mean by this? This neuron over here might be looking for an eye, so it's looking in this region, right? So all of these neurons are looking in this region. Then when I get to the next layer, this is looking for a small region in the output of the previous layer. So which means that the next layer of S cells is actually combining a bunch of these guys. And it actually responds to a much bigger region in the input. So as you go through the network, the receptive fields of each of these neurons actually gets larger and larger. And the patterns that they actually detect get more and more complex. Fukushima actually proposed an unsupervised mechanism by which these layers could be learned. So firstly, he said that, of course, as I mentioned, that only the S layers can be learned. And he proposed a Hebbian mechanism for learning. So consider a location over here. This is being looked at by all of these locations in the different S-planes. But this particular thing is either a nose or an eye or a mouth. It's not all of them at the same time, which means that this is going to cause a firing in only one of the S-planes, not all of them simultaneously, right? So for each location, you would start randomly, and for each location, you would find which of these guys fired most strongly. And then you would attribute that particular signal entirely to that one location which fired most strongly. And then you'd increase the weight using the standard Hebbian rule. So again, details are not relevant. You can follow the whole structure through the entire network. And if you do, you end up finding these very interesting detectors. So he showed, using a model of his, a computational implementation of his model, that if you train this system using lots and lots of characters, images of characters, for instance, then the S cells in the first layer might end up learning patterns of this kind. Then the next layer, they learn a slightly more complex patterns. By the time we get several layers deep, each of the cells is actually learning a fairly complex pattern. And this is completely unsupervised. There is no signal. All you're doing is using Hebbian learning, starting from a random start. And so here are some of his experiments. Here he presented digits. And he showed that after 3 layers, one of the complex layers learns to detect the digit 0. Another learns to detect the digit 1. And it can do so regardless of distortion in the image and noise in the image. So basically, this entire process has learned to cluster the input into categories automatically. And can you shut the laptop? Yeah. So, and does this quite nicely. Now, the semantic interpretation of it, of course, is unclear, right? What exactly do all of these structures that have been learned in an unsupervised manner mean? So for that, we need to have supervision. And how can we have supervision? The neocognitron is fully unsupervised, and the notion of adding supervision to it obviously comes up. There were several proposals. Homer Atlas and Marx in 1988, and in CMU, Lang and Kevin Lang came up with a very nice model in 1989. And that's what became time-delay neural networks. And a year later, Jan LeCun independently came up with the 2D version of what we call TDNNs in CMU. And that's what we call ConvNets. So what is the basic idea? Instead of simply letting these things learn all by themselves without any reference to truth, what you can do is then take all of this final layer complex outputs and pass them through an additional classifier, like a multi-layer perceptron. And this classifier can now have assigned labels, for example, digits. And once I do an assignment of labels, now I actually have some truth that I'm assigning to each image. I can use this information and pass it back and learn all of the parameters of the network. And so this is basically what, and this you can do using simple backpropagation. You still have the same structure. You have the S layers, which are looking at the outputs of the previous layers. You have the C layers, which are doing a cleanup of what the S layers do. And then you have S layers again, which are looking at the outputs of the previous C layer, and so on. But the output of the final C layer goes into this classifier, which decides, which takes a decision. And that decision can be validated using training data. Now backpropagation can learn all the parameters of the network. So now again, remember that when I have a pattern of this kind, in each of these planes, each plane has many cells, many neurons, all of which have exactly the same behavior, which means all of which have the same weights and the same activation. Everything over here is identical. This is like saying I have a plane of these cells together. They are looking at my entire input field. But that's actually no different than having just one of these cells which scans this input field to cause this map. Having many identical ones is the same as scanning with one and producing the output, right? So which means we only need to know the behavior of one. Now, in Fukushima's model, the receptive fields were elliptical. Jan LeCun changed those to rectangular, square. So S cells would have some KL cross KL receptive fields. C cells would have some other square receptive field, which is to say it's looking at small square patches in the output of the S cells. And so what exactly is each of these S cells computing? The S cell is now, it has if you look at what this guy computes from here, it's looking at some input region of the image. It's going to, and remember, our neurons are all correlation filters. You're looking for your weights represent a pattern that you're trying to detect, right? So which means that corresponding to each of these locations, it's going to have a weight. And it's going to compute the product of the weight over here and the pixel value at the same location. You're going to do this over the entire patch. You add them up. And that's the correlation between the input and the weight pattern, right? So, yes?
Do we assign an individual weight for each pixel?
Yes, and we will see, right? So within this block, just within this block, okay?
That's a lot.
Not really. Because, and so here's what happens. This is the weighted sum that you're looking within the square. But then in subsequent layers, because you're looking at many of these, many planes from the previous layer, you're also adding over all of the planes. And then finally, you have an activation. So that gives you the output of the simple cells. The complex cell is looking at a square region of the output of the simple planes, and it's just picking up the maximum. Right? Yeah.
So, what were their thoughts on the number of layers? So, did they have a fixed number of simple cell layers and complex cell layers? Or?
They just Fukushima simply had a hypothesis about the entire structure. In his model itself, computational implementation, I think there were 3 layers, 3 pairs of layers. But the actual numbers, they left that open. This was trying initially, if you go back to the biology, they were trying to explain the behavior of the mammalian vision. But then subsequently, these are just computational models with many things left open. Yes?
This model explains how we are detecting the structures or patterns that we already know of. What about unknown structures?
So here again, we don't. So here is the point. You don't really know anything. So if you look at what Fukushima's model did, all he did was he presented inputs, and all of these patterns, all of these parameters were learned using Hebbian learning. Like I said, you just initialize all of the weights randomly the first time around. And then you would see which of these things fired most strongly in response to a signal here. And you would attribute it entirely to that one guy, and then you would increase that weight. Right? And then you'd share the same weight across all the units in this plane. So simple Hebbian learning is going to keep modifying the weights after presenting a sufficient number of inputs. He finds that when you look at the third layer, then there's one C plane which only responds to the digit 0. The other one that only responds to the digit 1. It's completely unsupervised. So you don't know anything about what they're going to learn. You're still explaining, in some sense, mammalian, we learn to see, right? How does that happen? But then when you get to the computational model, now you actually put in an additional supervision. And again, everything is being learned. The only things you're providing are the input and the label. Everything in between is being learned. Right? So here's what the output of the C cell is, right? And here, this was Jan LeCun's original implementation of this model. And he actually this was the first real-time recognition system for handwritten digits. And I think the USPS, US Postal Service, used it for many years. So he still has this demo on his webpage. We call this LayNet, and this is version 5 of LayNet. You can see that as the input comes in, now observe that this is also position invariant, because you're scanning the entire input, and this is kind of explaining the grandmother neuron behavior, right? Now, so the story so far, the mammalian visual cortex consists of S cells, which capture oriented visual patterns, and C cells, which perform a majority vote over groups of S cells for robustness to noise and to jitter. So the neocognitron emulates this behavior with planar banks of S and C cells with identical responses to enable position invariance. And only the S cells are learned. C cells perform the equivalent of a max over groups of S cells, and unsupervised learning results in the detection of useful patterns. You basically end up performing some kind of clustering. LeCun's LayNet added external supervision to the neocognitrons. So here, the S planes of cells with identical responses are now modeled by a scan with a single neuron. There's no need for me to replicate the neuron so many times. I can repeat it by the computational equivalent by simply scanning with a single neuron. And C planes are emulated by cells that perform a max over groups of S cells. And this gives us a convolutional neural network. Now, if you go back to the previous class, this is exactly the same model that we finally ended up with, right? So what is the general architecture of a convolutional neural network? A ConvNet comprises convolutional and downsampling layers. Now I'm changing the name from S and C to con and downsampling. So this S cell, what we call the S planes, consist of neurons which scan the input and create a map, right? So this we're going to be calling the act of convolution. Then the C cells, the C layers, we're looking at patches of the S cells and picking the max. So as a result, the C cell output was smaller in size than the S cell output. The image reduced in size. There was a downsampling going on. It was not a uniform sampling. You were picking maxes over small groups, but there was a downsampling going on. So these are the convolutional layers, which comprise neurons that scan their input for patterns. And these are the downsampling layers, which perform max operations on groups of outputs from the convolutional layers. Now, in a computational paradigm, there's no need for us to be trying to pretend that we are following biology. So biologically, every conv layer was hypothesized to be followed by a downsampling layer. If I'm implementing it, I can have a sequence of conv layers and a max. You don't have sequences of max layers because that can be replaced by a single max, but the rest of it, I can have any number of conv layers before a single max, and I can alternate them in any order. And then when you finally get to the last downsampling layer, subsequently all of these outputs are put through a classifier, which means you take all of these maps, you can flatten them out into one giant vector, and then you put it through either a single softmax, as we saw in the last class, or I can put it through an entire MLP to get an output. So this is the entire general architecture of the convolutional network. So the convolutional layers are learnable. These have the neurons, which we will call our filters, that actually learn to respond. The downsampling layers, they have only a simple job. They're looking at everything that happened before, and they're picking a max within a specific region. There's nothing to be learned about picking a max. So the downsampling layers have a fixed response. They're not learnable. Questions? On Piazza? Right. So let's look at the convolutional layers. A convolutional layer comprises of a series of maps, as we just saw, corresponding to the S-planes in the neocognitron. They're variously called feature maps or activation maps. Now, each activation map has two components. First, you have one linear component which computes the weighted combination of everything that it sees in the previous layer. And then once it computes a weighted combination, subsequently you have an activation that's applied to it, which performs some nonlinear transform of this weighted combination. So this little plane, if you look really closely, it's actually two planes. The first plane is computed by linearly combining patterns from the previous layer, and the second plane is arrived at by applying a nonlinear transform to the first plane, right? So this one thing has actually got two subparts. Now let's look at the first guy, right? This one is obtained whereby through a nonlinear combination of everything, through a weighted combination of patterns from the previous layer. So to understand what is really going on, let's look at a single map, right? Now, again, every position here has contributions from all of these guys. But I'm going to be looking at the contribution of just this one term over here first, and then we'll look at the more generic picture, right? So let's assume that this figure, is this guy here. It's binary. It's all zeros and ones. In my example, it's just 5 cross 5, right? Now, at each location over here, this value here is computed by looking at some small region from the previous map. So that small region in my example is 3 by 3. And for each location in this region, as we saw, I'm going to have a weight. So in the regions that I'm scanning, I would have a pattern of weights of this kind, like 1, 0, 1, 0, 1, 0, 1, 0, 1 in my particular example. I might also have a bias, because now this is just a regular neuron, right? So what would I actually do? This is my filter. This green patch shows my original. I'm going to call this my filter now. So this is a filter. This green patch shows the image from the previous layer, the map. And when I impose this filter on top of the top left corner, the black numbers are the original numbers in the image, and the little numbers to the bottom right are the filter values. So what I would be doing is multiplying this value by the filter value. So it's 1 times 1. This value by the filter value plus 1 times 0. This is 1 times 1, 1 times 0, 1 times 1, 0 times 0, 0 times 1, 0 times 0, 1 times 1, right? And then I add all of them up. If I add all of these, so this is component-wise multiplies, all added up over the entire patch. And when I do that, I'm going to get one number, which would be this number 4 over here. And now there's the business of the scan. So you don't do this just once, you do the same thing at each location. And the output is now going to be the output obtained from the scanning, right? So this is what you get when you only, this is the response, this is the how one of these maps contributes to this location. But in reality, you're not looking at, and of course, when you're scanning forward, you don't need to go over just one pixel at a time. As we saw in the last class, you could be skipping multiple pixels. So if, for example, if here I was skipping 2 pixels at a time, that's going to be my output. It's only going to be 2 cross 2, right? Because at each time I'm skipping 2 pixels at a time. Scanning can be done at any granularity, right? But then let's look at what this business of scanning really means. You're not, when you compute the output value at any particular location, you're not looking at just one region. You're looking at all of these maps simultaneously. So what this means is that your filter is going to have, if I'm using a 3 cross 3 filter, that filter is going to have one 3 cross 3 pattern for each of these guys, and these patterns need not be identical. So here if I have m planes in the previous layer, and I'm looking at a 3 cross 3 filter, now 3 cross 3 is going to be 9 values. I have 9 values for each of these m planes, so the total number of values is going to be m times 9, right? And so what would happen here is I'm doing a component-wise multiply in each of these 9 locations, each of these 9 locations, each of these 9 locations, adding them all up to get this one value. And then I'm going to slide one forward and do the same, and I get the next value. Then I slide one forward more, I get the next value. And I can keep doing this to fill up this entire map over here. That's just one filter, right? Now I have a second filter, and the second filter is going to once again have 9 times m parameters, which are different from the first filter. And I can repeat the same operation, and I'm going to get a second map, right? And that's how I get all of these maps from these guys, right? Now, I can visualize this somewhat differently. Visualizing it like this may be a bit tedious, but instead of trying to keep track of what's happening in many things simultaneously, visualization is somewhat more convenient if I just take all of these guys and stack them up into one 3D object like so, right? If I stack all of these guys into one 3D object like so, then I can stack all of these 1, 2, 3 subcomponents of the filter, and the filter is also going to be one 3D object, right? Didn't get it? So just take, if I have, say, 3 of these over here, instead of keeping them one on top of the other, if I lay them one in front of the other, that just becomes a cube as opposed to many simple planes, right? The same thing with the filter, right? And so now this is a visualization of all of those planes simultaneously, and this is the visualization of the filter. And so what am I really doing when I compute the scan? I place this filter on this top left corner. I compute component-wise products, and I add the lot, and I'm going to get this one number. That's shown by this equation over here. I'm going over all of the planes. Within each plane, I'm going over the square, and I'm taking all of my weights, which are plane-specific and position-specific, and multiplying them with the corresponding y values, which again are plane-specific. This filter may have its own bias, and that's going to give me this value, right? And now I can, so this is from one map, that's just this one guy, and this whole summation is considering all of them, right? And now I'm going to keep scanning forward, and I fill up the entire map. This is my convolution operation. this business of taking component-wise products and summing them up at each location and scanning the input using this operation to produce a map is what I'll call a convolution. And so the computation of the convolutional map at any location is going to sum convolutional outputs at all planes, just to be clear, right? We saw how this is done in the last class. We actually wrote the code for this, right? Although what I've done over here is sort of start off from the biological perspective. Where did all of this magic come from? And it turns out that the magic didn't come from just taking a standard scan and inverting the operation. It started with the human eye. But regardless of whether you start with the human eye or from an engineering perspective, the eventual code and the eventual operations are going to be exactly the same. And so this is what you arrive at. Now, observe that each of my filters is a 3D tensor, right? The depth is the set of planes in the previous map, and the area is the size of the filter itself, width and dimension. So I have, within each layer, I'm going to have a bunch of filters. Each filter is now a 3D tensor. It's an object. And over the layers, I'm going to use, for each of my filters, I'm scanning the input. When I'm scanning the input, I'm basically slicing out the corresponding portion from the current outputs from the previous layer, and I'm performing a component-wise product between the filter and that block that I've sliced out, and then I'm adding all of the terms. Yes?
So your input is one photo with multiple layers, like different colors, right?
Pardon me?
So your input is one photo with multiple layers?
Yes, that for the overall network, but when you get to the second layer, now it's no longer a photo, right? But it's still going to have the same structure. So think of what happens. In the first layer, this would be a photo. Right. I mean, if I went back here, let me go back. So in the first layer, this would be a photo, correct? I have 3 colors. But now, from the photo, I have got many, many maps. So when I go to the second layer, now the depth is going to be as many as the number of maps I have computed. Now you can't really call it a photo anymore, right? So, clear to everybody? Yeah. And so, here's what we were really doing. Here is the operation we were performing. You were scanning for within each layer, for each filter, you were scanning the input. At each position, you are placing this cuboidal filter on the input and computing a component-wise product and then adding the lot, and that gave you the affine combination at one position. And you did this, you scanned using this operation, right? This is the operation that we first saw. Now again, the stride doesn't have to be 1 pixel. The output of the convolution, size of the output of the convolution depends on implementation factors like the size of the filters and the stride, but let's consider the stride in particular, right? So here, for example, the input maps are size 5 cross 5, the image is 3 cross 3, and the stride is 1. So how big is the output going to be? If you assume that the filter is not allowed to go past the edge, then here clearly there are only 3 skips before you run out of image, right? So the output is going to be 3 cross 3. Now if I had a stride of 2, after just 2 steps, I'm going to run out of image. So the output is going to be 2 cross 2, right? I can do the generic arithmetic. So if my input image is n cross n, and my filters are all m cross m, then what is the size of the output going to be? If I have a stride of 1, n minus m plus 1, right? And if I use a stride of s, it's just going to be divided by s. There's a flow ratio, right? So it's going to be n minus m over s plus 1. So very simple. So this, for a simple convolution, if you have an image of n cross n and a filter of m cross m and a stride of s, that's going to be the output size. So this results in a reduction in the output size. Even if s is 1, the size is going to reduce because you can't run off the edge. And this, in some cases, might be assumed to be unacceptable because, for example, suppose you're doing some processing of some input, and then after all of those convolutions, you want to look at what you get, and you want to think of it as being representative of the input. Then the fact that it's kind of lost size actually gets in the way of assigning input responsibility to each of the outputs, right? So sometimes you want the output to be the same size as the input, in which case we like to zero pad things, typically as symmetrically as possible. You add some columns of 0 to the right, some columns of 0 to the left, some at the top, some at the bottom. And you add just as many columns and rows of zeros as are required to make the output the same size as the input. The actual number, you can work this out, it's on the slides. So the convolution operation results in a convolution map, right? That's this guy. And then you apply an activation to it. The activation function can be anything. Typically, we use a ReLU. And so this is the same code as before. I just extended the yellow box to actually show you that once you compute the z's, you then perform the activations on it to compute the y's, right? So again, there's the downsampling operation that occurs between the layers. So the convolution and activation layers are followed intermittently by downsampling or pooling layers. What are these? They don't actually have to alternate. They can occur after any number of convolution blocks. So what are these? We've seen this before. This is, if you were looking at max pooling, you're going to be looking at a block of inputs and picking the largest value, and you'd scan forward all the way to the bottom. Now, this is just doing what our C layer was doing. It was cleaning up, right? But then it's also, the C layer was also sort of reducing the size. And the reason that happens is that when you do the max operation, you actually stride by more than 1 pixel. So this means that the output is actually going to shrink in size. So here, for example, if I were doing a max pooling on 2x2 and had a stride 2, this image is going to just be converted to this guy here. Observe that this conversion is non-linear because the location that's being picked over here can be very different from the location that's being picked over here or here or here. So this is a non-linear operation. Within each block, the transformation is different from the next block. So the max pooling operation is a non-linear operation. You can replace this non-linear max operation by the mean. The problem with the max operation is that when you're trying to learn stuff, you have to compute derivatives and pass them backward, and this becomes a bit of a challenge. If you compute the mean is a nice clean operation, and where you can pass derivatives forward. So sometimes we perform mean pooling, or you can do other kinds of operations like compute the p-norm of all of these guys. As p gets larger, this begins to approach the max more and more, or even replace the max with a learned network, right? And all of these have been proposed in the literature. As it turns out, we found lately, I'm not actually going to have pseudocode for the max operation on my slides, because in the latest architectures, we don't even bother with this pooling operation. There's no reason for us to be biologically inspired. We can be computationally inspired beyond a point. And if I'm simply convolving with a stride, I'm shrinking the outputs anyway. So, and the fact that we are using this distributed representation should naturally also give us some degree of robustness to local deformations and distortions. So it turns out the max pooling operation really isn't required. So which means I can get rid of the max pooling completely and just do the downsampling by a simple, regular convolutional layer, right? So here is a fully convolutional network without the max pooling. We've seen this before. You're going to go through layers. You start off with the image. Within each layer, for each filter, you're going to be scanning forward, and the scan may be in terms of strides, which may be layer dependent. At each position, you pick out a block from the three-dimensional structure that you had at the previous layer. Remember, I'm stacking all the planes into a 3D block, and then computing a component-wise product between this 3D block and your weights, summing them up, so you get the affine combination, then you apply an activation to it. So after having gone through all of the layers, the output of the final layer can now be passed. The collection of elements in the output of the final layer can now be passed either to an MLP or to a softmax or any other simple operation which gives you a scalar output, right? So the story so far, the convolutional neural network is a supervised version of a computational model of mammalian vision. It includes computational layers comprising learned filters that scan the outputs of the previous layer, downsampling layers that vote over groups of outputs from the conv layer. The convolution can change the size of the output, which you can control via zero padding. Downsampling layers may perform max, p-norms, or be learned downsampling networks. Regular convolutional layers with stride greater than 1 also perform downsampling. Right? Questions? Yeah.
I'm still not clear why the last bullet, I mean, regular with stride larger than 1 also perform.
That's because we saw this, right? I mean, way back when. So when I have a stride greater than 1, this shrank, even if I zero pad it. So if my convolution uses a stride greater than 1, the output is going to be smaller than the input.
Yeah.
So that's just downsampling, that's all.
Oh, but not pooling.
No, it does not perform pooling, it performs downsampling.
Yeah, because the size.
Yes. I didn't say pooling, right? Yeah. Yeah.
I actually got a question about pseudocode before the story so far.
This one? Yeah.
Yeah, this one. Yeah. Where is the summation across the different planes?
So this guy, I'm just calling this, so this Wij I explain as a 3D tensor, right? So it's got planes and filters. So I'm just calling this a tensor in a product, which is, I've got two tensors of exactly the same size, and I'm just summing them up. Right? Yeah.
So you perform max pooling, you perform on one feature map, right?
Pardon me?
You just perform on one feature map.
Yeah, max pooling is performed on one feature map at a time.
Perform on multiple feature maps.
You could, I mean, these are, at some point, these are engineering solutions. Considering that we don't even bother to pool these days, max pool these days, I don't know if trying to work on those is going to be very worthwhile, right? Yeah.
Today's modern architecture, do we still use the pooling layer?
Typically, we actually just get the size reduction through having a stride, the pooling layers. And the latest, at some point, we began having these all convolutional networks, which have a few more parameters. But it turns out that that's just as effective as having pooling, or sometimes even more. So we don't do it anymore. At least in most things that we will deal with, yes.
In the network, the planar network that we saw in the earlier slides,
Which one? This one?
the one before this,
Just go away. Tell me when to stop.
this one.
Okay.
The input to the first convolution layer, is this portions of an image or is it like the RGB layer?
So this, in the very first layer, this would be R, this would be G, this would be B, right? And we'll see that. I'm actually going to go through that in a little more, since I'm in the business of trying to explaining this. Let's actually go through this in some painful detail. And we'll also go through the business of derivation. I'm assuming I have the time, 20 minutes. So this class is probably going to leak over to the next class. Scott Fallman was going to be doing a lecture on the 18th. He will not be doing it. Instead, his lecture is going to be on the 27th. And so in the next class, I'll just try to do the spillover from the ConvNet, and we will maybe discuss a few other architectures and start off with recurrent networks in the subsequent lecture. Or, depending on what happens during this week's recitation, we may flip the order and start off with recurrent networks on Monday, and in which case I'm going to be postponing some discussion of convolutional networks to later. We will revisit the topic once we are done with attention, because it turns out that attention is actually also used with convolutional networks, right? Anyway, so questions so far? Yes?
So is there any particular intuition for why you might choose, say, max pooling over any other type? I mean,
so the original intuition was that it allowed for deformation, right? In the sense that if things jittered a bit, then you still wanted to say, I found this object, I found this pattern, not in precisely the location I was looking for, but just slightly off. And the notion of finding a pattern, as we saw, is saying that the detector for that pattern fired. Right? So remember, in the last lecture, we started off with saying, is there the word welcome in this recording? What we did was we looked for welcome everywhere and then looked at the max. That's just a max pooling. It's a detector, right? Position invariant detector, yeah.
So, but if you use a fully convolutional or a convolutional layer with stride, essentially, does it handle jitter?
It doesn't explicitly handle jitter. It just, but in exactly the same way that max pooling does, the operations are different. But the fact that we have distributed representations actually tends to naturally account for some degree of position invariance. So it doesn't account for jitter in the manner in which max pooling does it, but it does account for some jitter. Yeah. So let's go back to answer your question. How does the whole thing work, right? So let's say I was trying to perform classification of this image. Is there a house in the image, sort of thing? This is the grayscale. You could directly work off the grayscale, but it's color actually carries information. So now this has an R, a G, and a B. So the input is now going to be 3 pictures, which I can stack like so, right? Once I stack it, for the very first layer, we will typically work with square images. Our filters are also typically going to be square. Large networks are a problem, as you will see. Even no matter what the size of your GPU is, I can always give you an image which is larger than which causes you grief. So typically we will scale our inputs down to say size 128 cross 128, 64 cross 64, or in the case of the homework that you're going to be doing, 32 cross 32. It's scaled down just for computational purposes, so that you don't end up spending $3,000 on Amazon to get a core dump. So here's what your input is going to look like. And you're going to have a bunch of filters, say K1 filters. K1 is to say there are K1, the 1 is the index, which represents the layer. And each filter is going to have some span, physical span, which could be 3 cross 3 in my example. So now the input is going to be convolved with these K1 filters. Typically, the number of filters is going to be a power of 2, like 2, 4, 8, 16, 32. And another thing that we tend to do is to increase the number of filters as you go through the layers in powers of 2. So that's because as you go through the layers, the maps get smaller, but you try to extract more and more information out of them by using more and more layers and more filters. And the filter sizes, this L, so again, observe that the filter is L cross 3. This 3 represents the number of colors over here, right? What is L? We can use 5 cross 5, 3 cross 3, small. We want them to be small because the smaller they are, the finer the details that you're looking for in the image. If they're big, you're looking for coarse details, you may end up losing, and you lose a little bit in terms of jitter and position invariance. Or the filter can even be 1 cross 1, right? So you want these to be small enough to capture fine features, which is particularly important for scaled down images. But then you have this issue, what on earth is a 1 cross 1 filter? A 1 cross 1 filter is really just a simple MLP but this is a very interesting kind of MLP, right? It's going to go across the layers, but doesn't have any physical span. It's like an MLP which is going across the layers and scanning, or not an MLP, a simple perceptron, which is going across the layers and scanning the image. So to actually represent the filter, it's better to say 5 cross 5 and put the times 3 in parentheses, because the actual scanning is being done in the 5 cross 5 space. You're not scanning across the 3, right? So I'm just using this notation to distinguish between the scanned dimensions and the non-scanned dimensions. So the total number of parameters here are going to be the number of filters, the span of the filters, and the stride. So the total number of parameters is going to be K1 times here 3L squared plus the bias. The stride S is not actually a parameter, it's a design choice, right? Now, and you may also decide whether you want to zero pad it or not. So you have these K1 filters. What happens? Now, when I apply these K1 filters first, I'm basically going to be doing the scanning, which is given by these equations that we've seen earlier. You're summing over all the colors, and then at each position, you're sort of doing a component-wise product of the filter and the image, and you're computing a sum. I'm using a slightly different notation here than I did in the previous slides. I'm using a superscript subscript notation. The subscript represents the specific plane here. The superscript represents the layer. The reason I'm doing this is that otherwise the equation slides beyond the page, beyond the slide. So, and now you apply an activation on this affine combination. You'd get one of these guys for each one of my filters. You're going to get K1 maps because I have K1 filters, right? Now, so the total number of parameters, again, is going to be K1 times 3L squared plus 1. And all of these parameters must be learned. Subsequently, if you're actually performing a pooling, then you're going to be performing a pooling. Now, the pooling doesn't, this guy, you apply pooling on this separately to get this one, on this separately to get this one, and so on. And the pooling at each location is simply going to be finding the max of a small region from the input. Again, I won't spend a lot of time on pooling simply because it's really not, maybe the time is better spent elsewhere, right? But you get the idea. But if you actually implement the pooling, from each P cross P block of the map, that's going to be, you're going to pull down to a single value. And then you're going to be striding forward, right? So for pooling, the parameters to choose, the size of the pooling block and the pooling stride. And the choices, you have other choices. Do I choose max pooling? Do I choose mean pooling? Do I actually build a network? All of these are things to decide. Now, if you are performing max pooling, then you really want to perform this in 2 steps. You first want to find the location of the max within the block, and then read the value of the block at that location, simply because, so if I were trying to do a max pooling in this region, for this variable is going to say which of those locations held the largest value, and this is going to say what is the value over there. And it's important to retain both of these values separately, because both of them are going to be required when you're doing your back propagation to compute the derivatives. And so these would be the pool maps. And this is going to be K1 deep because you have one map per filter. So you're going to have K1 planes, right? So for the next layer, you're going to have filters again. You would have K2 filters. Each of the K filters is going to have 3 dimensions. One dimension is K1, which is the number of planes in the output of the previous layer. And the second dimension is the physical span, right? So the filters in the next layer are going to be K1 cross L2 cross L2. And if you apply those filters, you're eventually going to get, if you have K2 of these filters, you're going to get K2 such maps, right? Using the same operation. And then I can perform, so the number of parameters is going to be, observe, I have K2 filters. For each of these filters, each filter is going to be K1 deep and L2 squared in size. So it's K1 L2 squared plus the bias, right? So this is the number of parameters for the second layer. And then all these parameters must be learned. Subsequently, you can have a second pooling layer, and then you can keep, where again you have to decide the size of the pooling block and the stride. And then you can keep going forward. And finally, the output of the final layer is going to go through an MLP. And here again, you're going to have to decide how many layers, how they are connected, etc. Typically, we'll just use a simple softmax and be done with it. But in the early work, they used to have fairly complicated MLPs. So each convolution layer maintains the size of the image. If you have 0 padding, without it, it will shrink. Each convolution layer may increase the number of maps from the previous layer, because the number of maps depends on the number of filters. So even if the previous layer had only 3 planes, I can compute 100 filters, run 100 filters on it, and in the next layer now I'm going to have 100 planes, right? each pooling layer with hop D is going to decrease the size of the maps by a factor D. And filters within a layer must all be the same size, but sizes may vary with layer. And so also these strides. In general, in your designs, the number of convolutional filters is going to increase with the layer. So these are all the parameters you would have to choose. For each layer, the number of filters and the spatial extent of the filter, and maybe the stride. For the pooling layers, the spatial extent of the filter, the stride, and the type of pooling. That's something that you would decide. And for the final MLP, the number of layers and the number of neurons in each layer. Right? So here is Yann LeCun's original. I'm not going to get through this today. So here's Yann LeCun's original network. So when he was trying to do digit classification. So for digit classification, he had in the first layer 4 filters, each of which were 5 cross 5. So the images were 32 cross 32. So if the filter is 5 cross 5 and the image is 32 cross 32, the output is only going to be 28 cross 28. And you're going to get 4 of these. Then he had a 2 cross 2 downsampling where the stride was 2. So the downsampling makes this 28 cross 28 down to 14. Then he had 12 filters, each of these were 5 cross 5. So the 14 is going to go down to 14 cross 14 becomes a 10 cross 10 with 12 filters. And so you have 12 10 cross 10 images, which he then shrank down by max pooling to 5 cross 5. So you have 12 times 25. That is the total number of values you're going to compute in the second layer, which is what? 12 times 25 is 300, right? And then those 300 values were finally put through an MLP, whose size I don't remember, to give you the digit classification. Works quite nicely, still works very nicely, right?
May I know how to go from the red region to the blue region? Because the add part.
So this one here, you have 300 values. I can take those 300 values and arrange them as a vector.
Oh, just the range.
Just flatten them,
Yeah.
right? And then it's just regular MLP, right? So yeah.
Just assume the order.
The order doesn't matter thereafter. You just get rid of the order. Exactly. Right? So now learning the network, it turns out I'm going to have to spend some time about learn the back propagation, and I think there's no way I'm going to be able to do it in 8 minutes. So I'll let this leak into the next class. Right? Questions? Yes.
Two questions. One is probably relating to the training part. Suppose we are to use the max pooling.
Uh-huh.
Now, how do we do the back propagation through the max pooling layer?
I will cover this in the next class. Yes.
Actually, one more thing about the since we're having multiple filters within one layer, right? Is it very important that you have to initialize the different filters to different values?
Yes, because if they're all identical, this is regardless of whether it's a convolutional network or any other kind of network. If all the filters are initialized identically, then all of them will get identical derivatives. They will never diverge.
Yeah.
Yes.
So how to determine the value of k in each convolution?
So the number of filters. So the number of filters. This actually ends up being a little bit of a black art, right? And as in everything, it's hard to decide what the optimal number of parameters really is. And so the actual choices, the one that's most popular is to double the number of filters from between layers. And in the stuff that we've done, how many filters do we use in the first layer for your work? How many? No, in the first layer. So we start off, for example, when we do something like face recognition, which is in your homework. You start off with 4 filters in the first, 16 in the second, 64 in the third. But again, the point is, you want to, as the maps get smaller, you want to keep decreasing the size of the filters. Yes.
Can we intuitively understand how the structure, how the network retains the structure in the image? If we are doing face recognition, it could be just a picture with random combination of eyes and nose and mouth. How does it know that it's a face and not just a random?
So, well, obviously your classification output is going to tell you whether it's finding faces or not, right? But let's take a look at what the network actually learns. Here, this was on MNIST, and I should really have some labels over here. So we were looking at the receptive fields at the different layers. What is the pattern of inputs that the neuron actually responds to? And in the first layer, of course, it's just the weights. It's easy, right? But when you go to the next layer, that's actually more complex because it's looking at the responses of all the first layer neurons. So you have to sort of do some computation to figure out what is the input patterns, what are the input patterns that best fires the next layer. And you have to do this for each layer. But then if you look at what patterns the different layers discover are, here's what the first layer filters seem to discover. Then as you keep going deeper and deeper through the network, you can see that the individual filters begin to capture more and more digit-like patterns, right? And so as you go through the network, it learns, it literally sort of behaves in a manner that you would expect it to behave very often. In that if I'm trying to do a digit recognizer, you eventually end up learning one filter in the deep end that's really good at responding to ones, another that's really good at responding to twos, and so on. But trying to determine what these actual receptive fields are is a bit tricky, and it's actually a good exercise for you guys to go through. Yeah.
If we finally learned this, why don't we just find another 9 and do the correlation with the test data and say whether it's 9?
This is not exactly a 9, right? So the point is, it learns to, and so if if you look at what's really happening over here, across this, it's not that you have just 9 neurons in the final layer or penultimate layer. So you are probably going to find many different variants of 9 that it has discovered, all of which map to 9, right? So you can definitely go ahead and build a nearest neighbor classifier. This is a very trivial example of digits where nearest neighbor classifiers will do really nicely. And your best neural networks don't really very much outperform on MNIST all the regular classifiers based on standard approaches. But when you get to more complex tasks, it really becomes hard for you to come up with such clean interpretations. And there was a paper, for example, when I forget who it was, the guys at Google maybe, they built this large image classifier which was trying to recognize 1,000 types of images. And then they were very happy when they found that the receptive field of one of these neurons looked like a cat. And they published a paper in Nature or Science or something, right? So that's how, which gives you an idea of how challenging it is to actually find interpretations for what these intermediate patterns might be. Thank you.